we literally raised tent caterpillars in a 2-bedroom trailer. It was the Entomology Field Research Laboratory. We raised them at 85 degrees so that they would grow faster than anything in the field. And we took those caterpillars and threw them under the feet of those pregnant mares to increase the exposure to eastern tent caterpillars. And sure enough, in the space of a couple weeks, we started seeing very significant losses in the treated plots, the treated animals, and no losses or fewer losses in the untreated animals.
Welcome back to the Middle Tech Podcast. You've got Logan Jones here, and today we are diving into a Lexington-based biotech startup with the team from Lepidext. We sat down with the CEO, Bruce Webb, who is a professor emeritus and veteran virus researcher at the University of Kentucky, and Mary Wallace, a research scientist who are working to bring a new kind of pest control to market. So here's the problem. The corn earworm is one of the most destructive pests in U.S. agriculture, causing billions in crop losses each year. Lepidext is solving it not with chemicals, but with biology, using a naturally occurring virus to sterilize the pest population and protect crops without harming the environment. In this episode, we unpack the fascinating origin story of Lepidext, from a horse pregnancy crisis that sparked unexpected scientific discovery to a biotech startup born in Kentucky's ag tech ecosystem. You'll hear how the team navigated the regulatory maze of EPA approval, what makes this viral solution radically different from traditional pesticides, and how university research is being transformed into real-world impact. It's a story of science, grit, and a bold vision for the future of farming. Before we get into it, a word from our sponsors. Before highlighting our sponsors, we'd just like to state that the views and content shared on this platform do not necessarily reflect those of our show sponsors. Middle Tech is presented by KY Innovation, the Kentucky Cabinet for Economic Development's Office of Entrepreneurship. KY Innovation exists to support and develop Kentucky's startup ecosystem, and we are proud to be supported by an organization whose mission aligns so closely with ours. If you're a founder building in Kentucky, you need to check out the resources that KY Innovation has to offer. You can find more information by clicking the link in our show notes or going to kyinnovation.com. All right, Bruce, so when we talked on the intro call that we had, you told a story about how this research kind of came to be that eventually led to Lepidext, and it's such an interesting story, a big crisis that was in Kentucky in the horse industry. So talk a little bit about what was happening and how the research kind of ended up into Lepidext.
Yeah, Logan, that was just a really interesting time in my life. In 2001, we had an epidemic of equine abortions that was known as mare reproductive loss syndrome. And nobody knew what caused it. But in 2001, in the space of about 6 weeks, we lost 30% of the equine thoroughbred crop for that year. Hundreds of millions of dollars in 6 weeks, and nobody knew what caused it. One of the ideas that people came up with was that eastern tent caterpillars, which is a native insect that's here every year, somehow was involved in causing those equine abortions. My lab works on caterpillars and insect viruses, and so I was asked to investigate that. And that led to 2 years of really hard work where we were able to show that, yes, in fact, eastern tent caterpillars can cause mares to lose their foals. And we were able to figure out how that happened.
Yeah, and I feel like we just need to underscore how big of a crisis this probably was, because people who are in horse breeding, I mean, they're spending hundreds of thousands of dollars in some cases to have these horses breed and produce offspring. And for those offspring to be aborted, I'm sure that people are willing to throw whatever type of resources to solve that. And so your team comes in, you're essentially doing what sounds like investigative research in a way. So talk to us a little bit about what you discovered, like how the caterpillars were actually causing these fetuses to be aborted.
We talked to all sorts of farmers, and the first thing I did when I was asked to look into this was I went to 3 different farms and I walked the fields. And so I was looking at where the eastern tent caterpillars were, and they are found on cherry trees. And I'd look at a 30-acre field and I would see, one row would have cherry trees on it, and trying to figure out how that would happen. And then we talked to farmers. I talked to one farmer who said, I lost a foal if it had stood up, it would have been worth $2 million. And there were literally tears in his eyes, and not just because of the money. It's the effort and the time that goes into the selective breeding, and this is really the lifeblood for those people. And we took our system and just approached it analytically, and we said, okay, we need to manipulate the exposure of pregnant mares to tent caterpillars. Now, there is an entire industry of nurse mares that are used to, they're bred in other parts of Kentucky, brought to central Kentucky, and when a thoroughbred has a foal, they put the foal on the nurse mare so that the thoroughbred can be bred back again the next year. Those nurse mares were unemployed in 2002 because of the losses that we had, and we were able to rent the services of those nurse mares for our study. And we literally raised tent caterpillars in a 2-bedroom trailer. It was the entomology field research laboratory. We raised them at 85 degrees so that they would grow faster than anything in the field. And we took those caterpillars and threw them under the feet of those pregnant mares to increase the exposure to eastern tent caterpillars. And sure enough, in the space of a couple of weeks, we started seeing very significant losses in the treated plots, the treated animals, and no losses or fewer losses in the untreated animals. We didn't know how it worked, but in 2002, we were able to tell farmers, keep your pregnant mares away from tent caterpillars. And then 2003, we were able to propose a pathway for how this was happening.
And you said that was because of the spines on these caterpillars were hollow? How does that correlate? What happens when they eat them?
So the mares, and not just mares, but all horses, they're grazing and they ingest these caterpillars inadvertently. We don't think they select them, but they get a mouthful, and the spines on the hares are hollow. The spines on the caterpillar are hollow, and they penetrate the mucosa. So they, from the mouth to throughout the intestine, these pairs, it's essentially like sticking a straw through your skin, and it lets the bacteria that's in the gut invade the bloodstream. And then the bacteria circulates through the bloodstream to parts of the body that are immunocompromised, and that includes the fetus and fetal tissues, the placenta. And in those tissues, the bacteria was able to replicate without restriction, and that's what caused the abortion. I should also mention that this was clearly not all my work. This was a research team.
Sure,
we had microbiologists, we had equine reproductive specialists from the Gluck Center, we had pathologists, so it was a team effort.
And I'm sure it was very, very rewarding to probably be able to go to those farmers and say, we've figured out what's going on here. Here's the solution. I mean, for something that was as large of a crisis as it sounds like that was.
Yeah, you knew your work made a difference.
Yeah,
and that's what led to, in an indirect pathway to the founding of Lepidext with what we think is really a bigger and more important target than the thoroughbred industry.
And so talk us through that. You mentioned a bigger target and a bigger impact. Kind of talk us through the problem that you guys set out to solve with Lepidext, and then let's work our way towards how that research kind of came to be.
Yeah, let me introduce you to the corn earworm. So everybody knows the corn earworm because when they buy an ear of organic corn and they open it up and they see a worm on the end of it, that's the corn earworm. Okay? Now, the corn earworm attacks all kinds of plants, cotton, soybean, hemp, pistachio, peppers, tomato, lots of things. And they cause tremendous damage because they feed on the crop and on the most valuable parts of the crop. This insect in the U.S. causes $2 billion of damage or cost of control per year, and it causes about $10 billion globally. Not only that, but this insect it's essentially genetically predisposed to become resistant to insecticides. One of the things that really struck me was I read that 50% of the pesticides sprayed in India and China are to control this one insect. And so what we have is an alternative way to suppress the corn earworm population.
And I want to pause real quick. I think we might be picking some of them up. Are we okay?
I wasn't even hearing it.
Okay. All right. Just wanted to make sure we can cut that part out, but just want to make sure we weren't getting any
Yeah.
of their audio on out there.
I told them to shut up. No, I told them to stop making noise. Well, I'll quit dropping the tools on you.
Set them down gently.
Yeah, there's a reason.
All right. So 50% of the pesticide sprayed is trying to combat this corn earworm.
Yes.
Now you guys have developed a different take on that. So rather than using chemicals and pesticide to combat the corn earworm, you guys have developed research that takes a completely different approach to that. So how does this new mechanism of combating that pest work?
So what we do currently is we try to control the pest after it gets into our crop. And the problem is that these insects burrow into the sweet corn or whatever the fruit is, and the pesticides can't get to them. Okay? So once you have an infested crop, it's really, really hard to control these insects, which is why they spray so much pesticides, and it really doesn't work very well, and that leads to resistance. What we try to do is sterilize the moth. We prevent the eggs from happening by taking care of them, by preventing their production. And we found a virus that it has evolved from nature to sterilize these insects. And so we've just taken what nature has given us and made it work a little better for our purposes.
So you're just essentially taking a virus, you're figuring out how to express certain genes within that virus to have an outcome that leads to sterilization so that the pest cannot continue to go throughout the harvest and ruin the crop.
Actually, that's exactly right, but it's 180% wrong.
Okay.
So
turn me in the right direction.
Okay, so in the right direction. We're not adding anything. We're not changing the virus gene expression. We're taking away one gene.
Okay.
So think about, when you come to a fork in a road, there's a choice that the virus makes to either become what we call lytic and sterilizing, or latent and asymptomatic, and you're essentially become a carrier for that virus. Mm-hmm. Now the virus the virus plays the long game. It wants to stick around, forever. This virus is 60 million years old. Literally, from the time that dinosaurs walked the earth, this virus was infecting insects. So what the virus does is it prefers to go down that asymptomatic latent pathway because that's how it gets to the next generation. That's how it's inherited. What we did was knock out a gene that is required for the virus to go down the latent pathway. And that then forces the virus to go down the sterilizing pathway. It's terrible for the virus. It makes the virus sick, less efficient, doesn't work nearly as well. But it's great for us because it very efficiently sterilizes. The virus has all these mechanisms, to spread and control the populations, and we're just, accelerating that, to meet our purposes, to meet our ends.
I just find it so interesting to do research, understand a virus, be able to take that virus and use it as a tool, rather than just try to eradicate the virus or wipe it away or use pesticides to control this pest. You're now taking something that is naturally occurring and using it as a tool, and then building a business around that. And so this research has come out of the University of Kentucky. Mary, I wanted to shift to you really quick. You a product of the University of Kentucky and ended up joining the company. Would love to just hear a little bit about your background and what drew you to Lepidext in the first place.
Yeah, so I'm a native Kentuckian and decided to stick around. I got my undergraduate at Transylvania and then my PhD at University of Kentucky in entomology. And so Bruce was actually on my committee, so that's my kind of original connection to Lepidext. And my PhD research was also kind of in the biotech space, also looking at biological solutions for pest management, but more in forest systems. And I toured Lepidext for the first time around halfway through my degree and just saw that there was something going on here that I thought could really make a difference. And since my interest was already kind of going into industry, when graduation came last May, I was ready to join the team. And it's been a really awesome ride since.
Yeah, super cool. Well, I'd love to talk through kind of the life cycle of this virus as well. So just kind of getting more into the details of what happens once you release these moths that are infected with this virus into the field. Can you just kind of take us from there and walk us through what happens past that point?
Okay, so the virus replicates, again, only in the reproductive tissues. And what that means is that, all of the eggs and things required for making a fertile egg just get obliterated by virus infection. And on the female, it actually secretes what we call a plug, which is basically a secretion of pure virus that becomes a waxy pellet that you can see with your naked eye at the end of the female abdomen. So an uninfected male is searching for a female, finds her, attempts to mate with that, and acquires the virus infection, and then is able to spread that infection to the next female he encounters. Females, their biology is manipulated by the virus. The reason that we have runny noses and cough is not because it's good for us, but it's great for the virus to get around. So the virus in the moth, actually, it destroys all the reproductive tissues except for the pheromone gland. The pheromone gland in the female is actually hypertrophied. That means it's like 3 times bigger than it's supposed to be. The female produces 6 times more pheromone than an uninfected female and attracts males twice as efficiently as an uninfected female. And so that means the virus has evolved really just amazing ways to manipulate the biology of reproduction to facilitate its own getting around. We've all just lived through the COVID crisis. They have something called an R statistic, and that R statistic is how many people become infected from each infection. So COVID has an R of 3. Measles, which is one of the most infectious viruses known, has an R of 12. The R value for our virus is 10. So for every moth we release in the field, we infect 10 moths, if they're available to be found, in the field, which means that we can inoculate a field with a low number of insects and infect, pretty much everybody in the field.
And I'm sure for those listening, the next question is, okay, how do you contain this? How do you make sure this doesn't just spread unabated and wreak havoc in this kind of population of corn earworms? Maybe we don't want corn earworms at all, but I assume that they play some sort of role in the ecosystem. You don't want to just decimate their numbers. So how do you keep control of this? Like where does that part come into play?
Yeah, so something interesting about kind of the way the virus works is it's kind of self-containing in that because it's so infectious and so sterilizing that it's not able to continue into the next generations because all of those insects are sterile. And so kind of after the end of the season, there's no more of the virus spreading throughout the population. So essentially it kind of burns out on its own after kind of working throughout the season.
There's actually another factor too, because when we don't release these moths randomly. We release them into a sweet corn field that is in the stage called silk. And that is like the most attractive place for a corn earworm to be. And so when you release a moth into a sweet corn field in silk, it's not going anywhere. We put pheromone traps 50 feet off of the edge of a sweet corn field that we released moths into, and we didn't catch anybody. So it's about timing. It's not broadcasting. It's specific timing of the release, targeting the crop. The females are designed to find each other. The insects we release are designed to find each other. So there is, as Mary says, self-containment, both biologically and behaviorally.
I mean, from somebody who knows nothing about this sort of field, it really does sound like this is a perfect solution. It's naturally occurring. You're not spraying chemicals on the crops that we eventually consume. What's the catch here? What's counterintuitive about this? What are the cons? I mean, are there any cons, or is this truly like something that is just a much better solution than what we've had in the past?
I think it is truly something that's better than what we have in the past. There are, like any technology, there are limitations. And frankly, the studies that we have done are limited to small acreage by the EPA, by the Environmental Protection Agency. For an unregistered product, you have to do controlled studies, and we've complied with those very strictly. So we can't answer all those questions right now.
Yep.
But everything that we have done and everything that we have tested is strongly supported. This is not a chemical. You can't keep it for 2 years. You can't put it on a ship and send it across the country. You have to produce live insects and deliver them at the appropriate time. So a great deal of what Lepidext has done is really build very efficient productive systems so that we can produce enough insects to treat, an acre for, a few dollars.
I'm curious what it's been like going through the EPA process, because I'm sure that's been a very arduous process, and whatever you're able to talk to about it. First, just how long have you guys been developing this solution, this business, this research? I know it's been quite some time. So talk through that aspect of it, and then kind of where you are in this journey of working with the EPA.
So we had the idea for the project in 2016. We wrote a competitive grant proposal to the National Science Foundation that was funded, and that let us build the virus. We founded Lepidext in 2017 when that virus worked. And so we've been working on it since 2017. We started the EPA in about 2020, and we submitted our application in 2023. And so it's been under review for almost 2 years, and we've been told that they're going to give us a decision in July of this year.
So a big year for you guys.
This is a very big year for us.
And talk about just how rigorous that entire process is. I mean, you've submitted thousands of pages of documentation to them. I'm sure that, for people listening, just talk about how much is required to make sure that this is a safe and effective solution as opposed to the alternative.
Well, this is the Environmental Protection Agency, and we support their mission and their objectives. So we don't expect any sort of gifts. But we have tested this virus in honeybees. We've tested this virus in human cells. We have tested this virus in non-target insects. We have shown how long it is stable in the environment. Everything that we have done with the EPA has been supportive of registration. We have had a public comment period where the public can evaluate our submitted materials and comment. All the comments that we received were positive. There are some real problems with the corn earworm management that we feel very well poised to address. It's a rigorous process, and it should be. But everything that we've done is flying colors, and now we're just waiting for the green light, and let's go scale it.
And yeah, go ahead.
Oh, and thinking about the technology, it's coming from a virus that's already found in nature, and even the mutations that are in the virus that we're using could occur in nature on their own. So kind of like we mentioned earlier, they'd burn themselves out in the natural population. But I think having something that is kind of already out there, but kind of using it in a way that's more effective is also really good for, knowing that it's going to be good for the environment and not cause these issues.
I mean, I think it really is just like some of the most fascinating side of biotech is, taking something like I mentioned that's out in the natural world, naturally occurring, and figuring out how do we make this tool, how do we leverage this into something that's useful and you can build a business around. And I'm curious what a successful launch and what scaling looks like post getting regulatory approval from the EPA. I mean, where are you guys kind of aiming to take the company once that happens?
So the approach that we've taken is to work with farmers from the beginning. So we've done field trials at the University Research Farm at Spindletop, but we've also done field trials in Tennessee and Colorado. We have a project with the University of Hawaii. And so the farmers that we're working with are, they're the leading progressive farmers. And so they have seen what we're able to do in field trials on their farms. They are our beta testers. So when we have EPA approval, we want to do relatively small scale, maybe 30,000 acres in the first year of registration, which is projected to be next year, 2026. And then those farmers become our advocates and ambassadors. And, like many industries, but particularly farming, they're going to believe another farmer a lot more readily than they're going to believe us. And one of the things that Mary's leading our field trials this summer, and so one of the things that has really been critical to us is building relationships with those farmers. And so I thought, Scott, talk about Scott and what we're going to do there.
Yeah, so one of the big field trials we're planning for this summer is in the area around Memphis, Tennessee, with Scott Follin. So he's a soybean, corn, cotton farmer in that area, kind of in the Mississippi Delta, and or I guess it's not Mississippi Delta. What would you call that area?
It's in the state of Tennessee. It is on the Mississippi Delta.
Yes. Okay. Anyway, but kind of by the Mississippi River, and so kind of our thoughts for this summer are getting more of that efficacy data on the corn and being able to show these farmers that our product is going to be able to protect their crop. And then also looking, as we mentioned, it's not just corn earworm, it's cotton bollworm or soybean podworm, depending on what crop it's in. So looking at other crops and seeing, where our technology could fit into those systems as well.
I'm curious from a business perspective, how this company, so you mentioned that you've been funded through research grants up to this point, which sounds like a very long and arduous process, but also once you get there, you've proven out the research, now it's time to transition to more of a traditional business model. So just kind of talk us through, are you planning on being able to scale the company in sort of a bootstrap sort of way where you're not giving up equity? Is there a capital raise to this? How are you thinking about this from taking it from, okay, we've proven out the research, now we're transitioning to a business, now we're trying to scale that business. Kind of how do you think about those different steps?
Well, it's a small business, so you have to do multiple things at the same time.
Sure.
Right? So we have relied heavily on competitive research grants. We've gotten about $2.4 million of that kind of funding. We've also raised a pre-seed and a seed round of funding from investors, including the Bluegrass Angels, Ag Ventures Alliance, which is an ag venture firm. So we have investor funding, and there are some things you can't spend research dollars on. And
Sure.
so we have used those investor funds to build that company. We're at a point now where we're raising funds to do our product launch next year. And so we're in the middle of it, right now, talking to investors, and we think this could be the last round before we become profitable.
That's super exciting, and I'm sure also just feels great to kind of get to that point after, almost 10 years from doing the research and proving out the model and now kind of getting it to a viable business stage. Yes,
sometimes you pinch yourself because you don't know how you got here.
Yeah. So I'd love to, one of the reasons I wanted to have you guys on the podcast, aside from just how fascinating the research and product you guys are building is this was research that came out of the University of Kentucky. And so I'd love to just talk through the how it works when IP comes from the university and gets commercialized. We're close with Ian McClure's team and the commercialization office at the University of Kentucky. So you've chaired that board. You've been very close to that process this whole time. Just kind of talk through that entire thing. There's so many different questions we could dive into there, but I guess just at the highest level, how does commercialization of IP from a university work? What do they get out of it? How does that kind of unfold?
Okay, so first, professors, postdocs, graduate students that come up with something that is potentially valuable, and assuming that they recognize it as potentially valuable, they have a responsibility to disclose that to the University of Kentucky IP committee. The IP committee will look at that technology and determine whether or not the university will support patenting of that technology. So there is a obligation on that side. There's also an obligation more on the sort of moral, ethical side. Research dollars are not gifts to researchers. Society funds researchers because we come up with solutions to problems, just like we solved the MRLS problem because I was fortuitously predisposed with my knowledge base to help solve that problem. I feel as a scientist and professor that this is a real responsibility. We need to look for solutions to societal problems, not exclusively and not all the time, but we need to be aware of that. If you see that solution, then you submit to the IP committee. They evaluate the work. They decide if they're going to support it. And then you have a decision as a faculty member. Are you going to follow your technology, either within the university or outside the university? Or are you going to encourage and help the university to license that technology? Or are you just going to let it go and see what happens to it? With my experience and the stage of my career, I felt like I had to push this technology. That it was really a culmination of many years of work in my lab, my scientific interest. And I didn't think that this technology would be developed without me helping to put together a team and finding great members and giving them responsibility and encouraging their engagement, to make this happen. So there are, the university IP process is multifaceted and complicated, and everybody has decisions to make as they go along. And my decision was to spend, to change from an academic to a more entrepreneurial path, because I think it can make a difference.
It's super interesting when you think of the different paths that you can take to entrepreneurship and how you go about solving a problem. And when you think about the research side of things, I don't want to turn this into a political comment here, but you see a lot of funding being taken away from the NSF right now. And then you see people kind of pointing fingers like, oh, well, $200,000 to research about XYZ that they think is not leading towards anything. And I'd be curious on your perspective of, you were doing research, you said you were kind of an expert in caterpillars. It's like, who would have thought that would kind of go step by step all the way to now we have the solution to a kind of a biopesticide that is providing a ton of value. So just kind of connecting those dots from research to, I love what you said just about you have a responsibility to provide, taking that research, it's not a gift, and providing value. I mean, I'm just curious your take on that entire kind of flow. So
I'm a geneticist and a virologist. And so my training was in the 1980s. My wife worked for a company called Oncogene, which found some of the first tumor growth factors. For my postdoc, I worked with an insect virologist who developed something called the baculovirus expression vector system. That system was used by Novogene to come up with one of the COVID vaccines, and was commercialized and sold for that purpose. So I've been around entrepreneurship, just a very long time. And I just find it fascinating and engaging, but you can't predict. You can't predict what's going to work. What you can do is train people and encourage them to be aware. And the reason that we started working on this virus was because I just thought it was a fascinating virus. I think it's just really interesting. And I've often told undergraduate students thinking about working in my lab, it's like, if you ever wanted to study alien life, you should really study insects, because that's the closest thing we can get, to an alien.
I'm sure a lot of people would agree.
they do things differently, and it's important,
Well, I want to dig into some of the roles and the expertise within Lepidext. Before we get there, though, just to linger a little bit longer on commercializing IP through universities, you have the people that are doing research that are, typically have been in this field for a long portion of their lives. I'm curious your perspective on you were that person and you identified a problem and then kind of made that transition to be an entrepreneur. When you're talking to students that are coming and working in your lab, how would you advise them on, you are a researcher, yes, and this is kind of what you're supposed to be doing here, but how do you also kind of keep their radar up to see problems and to think of them entrepreneurially so that when that opportunity to transition into something that's commercially viable comes up, they can recognize that? What would be your advice to other researchers or faculty members about that?
when I talk to students, I would often talk to them about, what do you want to do with your life? And, I taught many years in the agricultural biotechnology, medical biotechnology program. So these are students that are, very highly capable, motivated. Most of them want to be professionals, doctors and dentists and pharmacists, et cetera. And I'm looking for the ones that, that are just interested. They just have that curiosity. And I would always support, whatever objective they wanted to achieve with their career. But I would also say, just, keep your eyes open. You can make a difference. And I'll often tell people, how many doctors do you think there are working on heart disease? And how many doctors do you think there are working on insect viruses? And where do you think you have the greatest potential to make a difference?
Yeah.
And it's you think it's sort of a big fish in a small pond story, but it's not entirely that way. It's like there's just more opportunity if you're working in an area that's important but neglected. And that's where we feel we are. And I think, that's what's attracted Mary to it.
Yeah.
Yeah.
Yeah. I mean, dig into that a little bit. So you saw what they were solving. You knew you'd done some work at the university level and you knew that kind of what interested you. But I mean, what other paths were you kind of considering? What would you tell, if you could go back in time, what would you tell your younger self about this path that you've now decided to take? Or what would you tell another student that's considering these kind of multiple paths?
Yeah, so I think kind of early on in research, it really just spoke to me kind of what Bruce was saying of like making a difference. And it kind of got to I had a few points that I was wondering, am I looking at this with rose tinted glasses? Like, am I thinking that I can do more out there than I really can? But, the more that I kind of went along in research and talked to more people in like internships or in like different industry positions, like I was really hearing all of this work that was being done and directly where it was getting applied. And so I think that's what really kind of drew me this direction. And specifically in this kind of startup ecosystem, it seems like you get to wear a lot of hats and you get to kind of try to push something forward that's new or kind of different from the like current like pest management or whatever field you're in. And so I think kind of being able to make that difference was the thing that really drew me this direction. And I think I would tell, a younger student, and I do tell students from other labs and things like, like there is an opportunity for you to kind of go from academia into a different role and still kind of keep learning and keep participating in that research, but get to make moves towards something bigger than yourself and something that you believe in.
I love that. And talk through a little bit more in more detail about your role here at Lepidext. So what kind of work are you doing on a day-to-day basis?
Yeah, so that really goes into the lot of hats comment. So I'm a research scientist, and so I'm working in R&D on different things. So some of it, one day, maybe we're working on improving our production system or ways that we can increase our moth production. And then another day, maybe it's writing one of those grants to the NSF or another one of the kind of funding agencies to work towards other ideas that we have moving forward with this technology. And then some days it's, planning on how we're going to conduct these field trials and talking to farmers and kind of figuring out what that's going to look like. So really every day is super different. And that's one of the things I really love about it.
Yeah, that's the startup world for sure. Bruce, talk us through a little bit from a broader picture, the different levels of expertise that you guys have that you've built on this team at Lepidext. So I know I got to sit in on a call with some of the rest of the members of the team. Talk us through kind of what fields are represented as far as the team goes.
So we have an executive team, and so the executive team, we all have PhDs in agriculture, 2 in the plant area, and I'm the entomologist. The other 2 PhDs don't do very much science. They contribute to lab meetings and discussions. One of them really functions as a COO and operates the business side of the company. The other operates more as a business development and functions in communications with investors and identifying opportunities for us in that regard. At the technical level, so this is the PhD level that Mary's at, we have 3 scientists there. As Mary, who just described herself as, a jack of all trades researcher, put my thumb in the dike sort of person.
Yep.
And then we have Megha Kalsi. And so Megha's actually working remotely. She's at the USDA Forest Pest Management Labs in Buzzards Bay, Massachusetts. That's at the base of Cape Cod. And the reason that she's working there is because she's able to work on a quarantine insect there, which can't be worked on anywhere else in the country. And we have USDA Farm Bill support for her project. And the idea is that this is an insect that's going to invade the U.S. It's a close sibling of the insect we work on. And we've shown that the virus that we're working on will sterilize this soon-to-be invasive insect. So, and then the third person, Jimmy Maines, is much more involved in production. He has, again, a PhD degree, but he's also the master and leader of our 5 3D printers, where we manufacture, design all of the insect rearing equipment to really revolutionize how many insects we can produce at a rapid speed and with the least effort possible. Then we have sort of a next tier of people that are supportive. Some of them are PhDs and some are more technical. We have Michael Anderson, who's a Iraq, Afghanistan war vet. He's been with me for the longest time, and he really does most of the insect production, particularly of sterile insects. We have 2 consultants, Daniel Webb, who does modeling for us, and Ellie Derbyshire, who's a mechanical engineer, and she works closely with Jimmy. And then we have people that are more directly hands-on in the insect production staff, Alex Adkins and Aslan Blevins. And so that's kind of the different tiers of staff that we have. One thing that I think is unique, and I think Mary will echo this, is that we're a very flat team. And we value contributions from everybody, and we value everybody. So there's a lot of the place is built on respect. It's built on inclusiveness. It's built on, finding a big problem and working hard to crack it. And we recognize that we need different kinds of people to do that.
I love that. Well, let's talk for a little bit about kind of what the bigger picture is here. So talk about how this could change how we farm the next 5 to 10 years. Are there other applications of this type of approach? Let's kind of zoom out for a little bit.
Yeah, so I think that kind of when you think about chemical insecticides and how they're used today, it's kind of a very reactive approach, whereas what we're suggesting is more of a proactive approach. So can we prevent these pest outbreaks in the first place and prevent the damage that way? And so kind of being able to move forward with a technology like this that's super species specific and doesn't have these negative impacts on pollinators or specifically predators, because in a lot of these cases, when you spray an insecticide, it kills off natural predators and leads to outbreaks of even other insects than the one you're trying to control. So being able to move towards this more proactive approach should hopefully allow us to really move away from the reliance on chemical insecticides when resistance arises and be able to, get ahead of the pest problem.
So it sounds like this is more broadly applicable than just corn or the corn earworm. I know your research is focused primarily on the corn earworm, but I mean, how broad can this get?
The biggest problem that I have is I want to spread out and tackle all of the different things that this kind of approach could address. We know that there are other viruses like this and other key pests that are great targets. We know that this conceptual approach can extend even further beyond that. So the problem is that I need to, take off my academic glasses and put on my, CEO business glasses and get a product to market. Mm-hmm. And then once we get this product to market, once we see its effect in the field, once we've proven that, then we can believe in it and start directing resources. We already believe in it, but then others will believe in it.
Yeah. Yep.
And then we can start looking at these other opportunities. Now, we are looking at those opportunities proactively and taking the steps that we can to protect the IP in those areas.
Sure. Yeah, I'm curious from your perspective as a founder, as a CEO, what parts of this kind of keep you up at night? I mean, what either gets you really excited or makes you nervous about kind of the road ahead for the company?
I don't know what the percentage is, but it's a very high percentage of business startups fail because they run out of money. Okay, so the thing that keeps me up at night is making sure that we have enough money to keep the great team we have going, and that's my primary job. EPA registration is something that a lot of people worry about. I don't worry about getting registration, I worry about the timing. If we have a 6-month delay from now, that has virtually no impact on us because it doesn't impact our field season. If we have a 12 to 18-month delay from our projected July 2025 decision date, then that costs us another year. And so right now, what keeps me up at night is how can I secure enough funding that we can survive a 2-year lapse in the event that there is some unforeseen holdup. So that's my biggest fear.
And what are you pitching to investors when you're going to raise capital in terms of the overall market here? Are you saying that this is a global market that you all can go and start really tackling, or how are you positioning yourselves when you're raising capital?
So it is absolutely a global market. We are not going to do a global market. What we are going to do is find companies in Brazil that are already using biologicals, license the technology into Brazil. We'll probably need to partner with them initially, do some field trials, prove the technology in Brazil, and then they will buy in. But the whole startup thing is the idea of a founder having a great idea and doing everything by himself. Well, that's just crazy. I mean, you need to build a team. You need to build collaborators. You need to reach out. You need to build a network. You need to rely on every resource that you can beg, borrow, not steal, so that you can achieve your goal.
Yeah, well, I mean, it's a completely different dynamic for a company that is very research heavy, very biology process oriented, like in the physical world. It's one thing to talk about software and scaling to a global market because there's no scaling cost really. You build the software once, you can give it to whoever. But for a company like this, I think you're absolutely right. I mean, there's just a lot of operating expenses and operating costs that go into scaling a company like this.
One of the things we've been very cognizant of is we don't have a lot of infrastructure costs. We think that we can build our production capability into a shipping container and build mobile production units and ship them to places where we're going to deliver large numbers of the product. So one of the things that we're actively talking about is enlisting farmers to test this idea, probably not next year, but in 2027. That would have the idea of being able to do a sort of franchise model where we're providing part of the biological infrastructure they need, but the actual hands and the production and the sales could be franchised into different regions. So we can get that farmer that we're collaborating with a new business opportunity that he could then market and sell to his collaborating farmers. So, this is a different kind of technology. They're just different opportunities than you would get with a classical, chemical pesticide.
Well, it's been a super interesting conversation. I feel like I've learned a ton. For anybody, I know we'll definitely want to continue to stay updated as you guys kind of go through this big year and see where everything takes you over the coming years. But for anybody that wants to keep up with you guys, is there a website that they can go check out, social channels that they can follow along? Just kind of plug any way that the audience can keep up with the work you guys are doing. Yeah,
so we have Lepidext.com. That'll kind of have our team and our story and anything you want to see about that. And then if you want to follow us on LinkedIn, that's where we're probably planning on keeping up with people the most. So
Awesome. Well, thank you guys so much for coming on. Super interesting conversation.
Thanks
Thank
for
you,
having us.
Logan. It's been a pleasure.